RNA interference, or RNAi, is an evolutionarily conserved mechanism of post-transcriptional gene regulation that uses an Argonaute protein programmed with a small RNA to downregulate specific messenger RNAs in a sequence-specific manner, leading to a decrease in their corresponding protein. You might have heard of RNAi, or even used it yourself in the lab, in the context of introducing siRNAs, in order to quote-unquote knock down the expression of specific genes to determine their function. Even if you've never heard of RNAi, let alone use it experimentally, your body is using it constantly, relying on microRNA-mediated RNAi in order to regulate the messenger RNAs of the majority of your genes. From cells in a petri dish to the trillions of cells in your body to, increasingly, vials behind the pharmacy counter, RNAi is a crucial regulatory mechanism. Let me tell you a little bit more about Argonaute and the workflow it has to go through. AGO is a powerful protein, but it can't act alone. Instead, it gets programmed by binding to a small RNA, which can be a microRNA or an siRNA, to form a core RNA-induced silencing complex, or RISC. These small RNAs are around 22 nucleotides long, and at their 5' end they contain a 6 to 8 nucleotide long seed sequence that allows AGO to use them as guides to find and bind messenger RNA targets that contain complementarity to the seed and variable amounts of downstream complementarity. These sites are typically located in the 3' UTR of the messenger RNAs, and upon binding, AGO is able to, alone or with the help of cofactors, induce translational repression and/or mRNA degradation in ways that I will detail in a minute. These diverse pathways lead to less of the corresponding protein being made, and once AGO releases the target, the RISC complex can then do this again and again because that core RISC complex, AGO plus guide, is incredibly stable. This gives a single RISC a lot of power to target messenger RNAs containing complementarity to that small RNA. So where do these small RNAs come from? MicroRNAs come from the transcription of long precursor products. They're transcribed in the nucleus as primary microRNAs, which get cleaved by a microprocessor complex to create a long RNA hairpin pre-microRNA that gets exported into the cytoplasm, where it binds to a protein called dicer. This is also where exogenous double-stranded RNA can come in to serve as siRNA precursors. Plants and invertebrates use viral double-stranded RNA to make antiviral siRNAs. However, our bodies have evolved more complex immune systems, and so we don't widely use siRNA in this way. Be it siRNA precursors or these pre-microRNAs, they can both get loaded into dicer, and then dicer is going to act as a sort of molecular ruler and scissors in one. It measures off a roughly 22 nucleotide RNA duplex, which gets loaded into Argonaute to form the pre-RISC. AGO then ejects the passenger strand, displaying the seed region of the guide strand for seeking out targets that match. Upon binding to one of these targets, what happens next depends in part on the Argonaute protein and in part on the amount of complementarity between the guide and the target. Some AGOs, including human AGO2, have what we call slicing ability. This means that if they bind to a perfectly complementary target, they can cleave the target across from nucleotides 10 and 11 of the guide in a process we call slicing. This exposes free ends of the RNA to exonuclease chewing, leading to mRNA degradation. With partially complementary targets, such as typical of animal microRNAs, AGO can't slice the target and instead has to recruit cofactors. It does this with the help of a scaffolding protein called GW182 that brings in decapping and deadenylation complexes to deprotect the ends of the messenger RNA and initiate conventional mRNA decay pathways. It can also interact with factors to induce translational inhibition and can sequester messenger RNAs. Once this target is released, the RISC complex can do this again. At the heart of these diverse processes is AGO. AGO has 4 main domains: N, PAS, mid, and PWE, as well as 2 linker regions, L1 and L2. The small RNA, so the microRNA or SiRNA, in this case miR20, went through Argonaute, with the 5' end held tightly in a pocket created by the mid and PWE domains, and the 3' ends held in the PAS domain. You can think of AGO as looking a bit like a duck. In this analogy, the PAS domain is the head of the duck, and its beak is biting down tightly on the 3' ends. The central region of the guide is disordered in this crystal structure, as well as other crystal structures. However, it is wending through Argonaute in an RNA binding channel, you can think of the wing of the duck kind of protecting the RNA as it goes through.